Okay, let's get rolling here. Hi, everyone. Welcome to Agrifood Conversations, brought to you by iSelect, the Van Trump Report, the Yield Lab Institute, and Family Farms Group. My name is David. I'm an associate here on the iSelect Ventures team. I'm excited to welcome you to the discussion today. Agrifood Conversations is all about driving innovation in agriculture. Each month, we highlight a specific theme, including emerging topics such as soil health, plant genetics, vertical farming, and aquaculture, to name a few. This month's theme is crop protection, and on today's call, we are joined by Jamie Batcher, CEO and co-founder of Boost Biomes. Boost Biomes is developing novel microbial crop protection products with improved reliability and effectiveness relative to microbial products developed using traditional approaches. Its proprietary discovery platform uses high-throughput DNA sequencing, selective enrichment, and advanced informatics to identify microbial products with important commercial roles. This approach is faster and less expensive than traditional approaches and uniquely identifies multi-microbial products, which are often more effective than single microbe products. Its first products will manage diseases of high-value crops, increase overall yield, and extend the shelf life of fruit and vegetables after harvest. Each of you knows that companies are more likely to succeed with the right network of customers, talent, investors, and advisors. We have invited you to this call because you're some of the smartest and most talented people in Boost Biomes market. You're potential customers for Boost Biomes products and services. You have built a company similar to Boost Biomes, or you are a sophisticated business cultural professional who understands the market and the challenges and opportunities Boost Biomes may face. Before we get started, we have a quick poll question to get a better idea of who we have on the call today. Please take a few seconds to answer. And while we're doing that, a few process comments while this poll is going. We are not soliciting investment. This presentation is to provide information to help Boost Biomes find customers, mentors, and other strategic relationships that can help them grow their business. Secondly, you are on mute. You can use the chat window to ask a question at any time. Thirdly, this presentation is being recorded and will be available for replay. And so with that, and without further delay, I am pleased to introduce Jamie, CEO and co-founder of Boost Biomes. Jamie, feel free to take it away.
All right, well, thank you very much, David, and thanks to all of the iSelect team for having me here today. I really appreciate it. And thanks to all of you out there. I'm really excited to get a chance to tell you a little bit about what Boost is doing. Let's see, there we go. So iSelect asked me to make sure that everyone knows that investing has risks. So we've added this. And if you'd like to contact the iSelect Fund, the Van Trump Report, or the Yield Lab Institute, here's their contact information. With that, let me switch gears and I'll talk to you a little bit about what we do at Boost and where we're heading. So as it says here, Boost is developing agricultural inputs. Largely our focus at the start has been in crop protection, in biofungicides. Underlying the products that we're developing is a microbiome discovery platform for understanding how microbes interact. We use that platform to identify product candidates, and then we go on and discover and develop new products from there. So let me jump in. I'm going to start with an example of what one of our products does. These are raspberries. We've treated or left untreated some of these raspberries, and this is on the first day. After we wait a few days, basically we see that untreated raspberries end up covered in fungus. Treated raspberries still look delicious, and you'd still be pretty excited and pretty happy to eat them. This is sort of the very personal version of the global problems that we face with agriculture. Partly with fungus, with fungal pathogens that destroy our fruit, but also other means that destroy food or waste food as well. So overall, I'm going to start with sort of a really big picture view here. That overall, about 6.5 billion people get fed pretty reasonably well. About 1 billion people go hungry every year. And yet we are wasting about 1/3 of the food we produce globally. If only that food was in the right place at the right time, we'd eliminate world hunger and we'd have a surplus. Of course, that's not what happens. And so people still go hungry. Wasted food also contributes to carbon dioxide into the air, all sorts of waste packaging. So there's really a lot of reasons to try and address this problem. Now, we waste, like I said, about 1/3 of the food we produce globally. In the US, we waste over 50% of the fruits and vegetables that we produce. Now, this isn't all because of fungal pathogens. Fungi play a role in that for sure. So let me be even a little more specific, and now thinking about it in the field, where fungi destroy, every year, fungi destroy enough food to feed 600 million people. And that's just in the top 5 crops alone. So addressing these kinds of issues is what Boost Biomes is all about. We think that there's a tremendous market pull for new products in the space. In other words, chemicals aren't doing the job, but there's also good reason for a switch from chemicals to biologicals. 2 of those reasons have to do with legislation and consumer preference. And I see these as being 2 sides of the same coin. Legislation is tending to make chemical fungicides less attractive to the growers. So there are things like re-entry times, times to harvest that limit the usefulness of chemical fungicides. There's also things like buffer zones, and in some cases, for some of these chemical fungicides, absolute bans going into place. On the flip side of that coin is consumer preference. We sort of very globally, we are thinking more and more about what's on the food we eat, what's on the food we put into our bodies. And very generally, consumers are becoming more thoughtful about wanting less chemical residues on the fruits and vegetables they eat. It's to the point where people track the dirty dozen and the clean 15, the fruits and vegetables with the most or the least residual chemicals on them. Now, I think of these all being related, right? That you've got the consumer preference on the one hand, legislation on the other, and of course, it's the consumers who vote people into office to form legislation. And so in the end, the growers are thinking about, on the one hand, what's the legal or regulatory attractiveness of chemicals? On the other hand, how can they best serve their ultimate end consumers? And of course, the business concern in the middle is, what's the cost and effectiveness of those products? We think that this is one of the places where Boost has a significant opportunity. I'm going to add to all of that with how, again, generally, we are becoming very thoughtful about how we can use technology to reduce food waste. Boost clearly sits in the prevention side of that. And this is expected to have very real business implications. So right now, today, the fungicides market globally is a $12 billion market. Out of that 12 billion, 1 billion is represented by biofungicides. However, bio and chemical fungicides are growing at very, very different rates. So within a decade, we're expecting that that bio will have grown by about 4.5 times, but the total market will have grown by only about 2/3. So there's an explosive growth going on here for bio, and bio is eating chemicals lunch. And we think that the landscape of how the majors in the industry are seeing this and reacting to it, we think that that is very supportive of what Boost is doing as well. I'm going to give just 3 examples here. Bayer has their LEAPS program, where they've invested over $800 million in 10 different areas. 2 of those would have been perfectly appropriate areas, or are perfectly appropriate areas, where Boost slots in beautifully. Syngenta, I'm going to give a less quantitative example, but the global head of biologics and their head of corporate venture is the same person. That, I think, says something very strategic about what they're doing. Not to mention that I think it was just last week they announced a very, very large effort towards reducing chemical usage. And then finally, UPL last year bought Arista, effectively creating for themselves a life sciences division for over $4 billion. So these are the kinds of quantitative and qualitative and strategic moves that the big companies are making in the space that we think bodes very well for companies like Boost. Now, we're going to shift gears a little bit. That was a lot of context. We're going to shift gears a little bit now to discuss, what does Boost do and how do we do it and what's different, what's exciting here? Well, our underlying technology is about understanding how do microbes work together in ecosystems. Now, this is incredibly important because we tend to think of of agriculture as something about biology, right? We've got plants are biology. But in fact, of course, it's ecology. There's a lot more physical and biological aspects to agriculture than just the crop itself. The physical side, you've got what kind of soil you have, what mix of soils, pH, temperature, amount of precipitation, many additional factors. And on the biology side, you've got the microbial ecosystem and the macro ecosystem as well that goes into what is the environment for any of the crops that we're growing. So with all of that thinking about ecosystems rather than biology, Boost's underlying technology helps us to understand, allows us to understand, how do microbes interact with each other? Now, first of all, we think that there's a lot of benefits to having products where the microbes actually interact. When you've got multiple microbes in a product and they're actually working together, they can help each other to last longer. They can have multiple modes of action to reduce the efficacy or to increase the efficacy of the product, to reduce the effectiveness of the pathogen. And they can also reduce the risk that the pathogen evolves resistance to the treatment. This is the reason why farmers usually use integrated pest management, or IPM, is by rotating chemical products, they get multiple modes of action and they lower the risk of resistance. Boost's products do that innately by having multiple microbes as part of the product. The approach is highly efficient and very effective. To give just some rough examples, if we compare with a company that's highly automated the in vitro screening process, first of all, that company would need isolated microbes, which is a lot of work, especially to build a very large library of microbes. They might screen hundreds of thousands of microbes in vitro, but ultimately find only one or a small number of microbes that might actually be effective as products. Another company in the space might use a more deliberate approach, systematically testing in small-scale fermentations. Is any given identified and isolated microbe effective at making what they might want? Well, they can find multiple microbes that way and develop a series of products that way. I'm using some rough numbers here to give you a sense of success rates, 1x, 16x. With Boost's platform, first of all, we don't work off of isolated microbes, we work off of microbiomes, the full collection of microbes from a given environment. So we can customize that group of input material for any given product that we seek to develop. That's a huge advantage with starting, but then also I'll give the numbers from a real example that we've done. With an input of only 4,000 different strains, we found microbes that work together. We found in the teens of microbes that we wanted to screen in vitro, about 8 that went into field trials, and 3 of those that still seemed effective after our field trials. Of course, we're pursuing the #1 most effective of those products, but 3 out of 4,000, that's 75x when you normalize it to compare to these other companies in the space. And finally, one of the key benefits of this platform is it explicitly takes into account the ecological basis of what the microbes are doing. Any other approach out there would have to take a factorial approach, which quickly becomes impossible to do in a lab. Factorial approach, just a quick reminder for everyone, is all the numbers multiplied together from that number down. So 10 factorial, 10 times 9 times 8 times 7, etc. When the numbers are in the thousands, those numbers quickly become astronomical. I'd like to give one example of what does our product do in a field. So I'm giving you some real field trial data of our active ingredient. This is a field trial done in grapes with botrytis or gray mold, one of the very common pathogens that grapes face. What you're looking at on the vertical axis here is the incidence of disease multiplied by the severity. So a measure of how frequently do the plants have it, and if they have it, how severe is the disease. UTC represents the untreated control. You can see that that's got a very high measure of disease. When treated with normal IPM, chemical products dramatically reduce the incidence and severity of disease. The boost active ingredient, unformulated and not used as part of an IPM, still also dramatically reduces the incidence and severity of the disease. So, like I said, showing you here data for botrytis, we have similar data on powdery mildew and downy mildew in grapes, and some similar data in other crops as well. I showed you earlier the visual representation of this on raspberries. We've also done a fair amount of work showing that the same kinds of results happen in post-harvest studies. So I'm going to move on from the technology and the science side of things to talk about Boost as a business. So in the middle of this slide, that's Boost. Those circles and lines are meant to represent Boost's technology platform. We sit in the middle of these sets of relationships where we do the discovery and the early development of our product. We work with manufacturers to make the product by fermentation, prepare it for sale with formulation and packaging. And then we work with distributors to get the product to the growers, to the packagers, so that they can use it in their fields, in their packing houses, and get the product out to the end users, to the consumers. We also have a variant of our business model, which is to co-develop products with partners, where a partner might come to us, identify a gap in their portfolio, and then we work very closely with them, and we can find alternate ways of commercializing the product that are, that make sense for both parties. I'm going to give an example here of how we're bringing our first product to market. First, we're partnering with a manufacturing partner and a distribution partner on this particular product. I can't say names yet because we haven't announced these deals, but suffice to say the manufacturer this isn't a contract manufacturer, this is a true partner. This is a global scale company. They make and sell billions, with a B, billions of dollars worth of microbial products every year. So they'll do the fermentation. They've already got great fermentation processes. They're working now on formulation, and they'll work with our distribution partner to get the product to the end users in the United States for high-value crops, high-value fruits and vegetables. And the distributor also, that's a true partner, one of the leading distributors in the US. And I want to give an example here. This is the very visceral demonstration of our partners working on this project. You can see that small tractor trailer, and you can see the mist coming out of it, spraying those apple trees. This is actually a field trial done by the two partners. So the manufacturer made the product in their fermenters, shipped it to the distributor. The distributor ran this field trial in the apple orchards. So you can basically see that the project has legs. The customers, the partners see this product as having legs, and they're running with it. I'm going to add to all of that by saying that was the first commercial arrangement for this product. We are now seeking additional distribution agreements for in the US, outside of scope of the first agreement, and rest of world for all crops. The wonderful thing here is we still will be working with the same wonderful manufacturing partner. And so they'll bring to bear their expertise at making the product. We'll get to work directly with the distributors for other areas, other regions, other crops, and also for the post-harvest opportunity as well. I'd like to give a bit of background of where we've been, where we are, where we're headed. I think you've gotten a good sense now of the snapshot where we are, but I think also seeing some of the trajectory of the company is going to be helpful as well. So Boost has been around now for 3.5 years. What I'm trying to show here is both the focus of effort and the corporate strategy, where the effort was around discovery or building a pipeline, getting our first product. That was early 2017. we did product development and over the course of basically the remaining time of the company from late 2017 on. And that's through until now. When now we've secured these partners, and this leads us to where we're going. Today, we're going to continue working with these partners to get the product out to the market. And as I just described earlier, working with additional partners to get this everywhere in the market we need it to go. But we are bulking up on our pipeline now. I'm going to talk a little bit more about this in the coming slides, but suffice to say, we've announced our first co-development agreement with Yara, the international global plant nutrition company. We're working on additional opportunities for joint development agreements. And in the end, we're going to have a pipeline of 3 internal products and 1 to 3 additional joint development agreements while we continue working on the platform. And all of this comes on the strength of a Series A round and a Series B round this year and next year. So we've been talking about the antifungal applications. And antifungals, we think is a very, very attractive space for Boost to develop our first products. But by no means is this the first or only challenge in the agriculture space. So we could also work in antibacterials, and the approach we would take from a technical perspective would be almost identical to generating new antifungals. We can work in the fertilizer space, collectively over $100 billion markets. And with some additional technology and infrastructure, we can also get into the herbicides and insecticides spaces as well. And of course, the reason we can do this is because we do have a platform technology. Microbiomes come in, products come out. And like I mentioned, I'm emphasizing here, but like I mentioned, we've got our first partnership secured with Yara in the phosphate fertilizer space. Boost is backed by a wonderful, maybe a little bit eclectic, but a wonderful group of investors. Yara is one of our major investors. We've also been invested by Y Combinator, by Viking, Kureha, the Japanese chemicals company, where one of their divisions is in agrochemicals, Cavallo Ventures, the venture arm of Wilbur Ellis. We also participated in the Thrive Accelerator and the Illumina Accelerator as well. So we've got a perhaps eclectic, but really a wonderful, diverse, and supportive group of investors. To quickly introduce to you the team, I'm Jamie Batcher, the CEO and co-founder. Been in biotech now for over a dozen years after having gotten a PhD and done a couple of postdocs. James Pierce is our Chief Scientific Officer. We brought him on from Monsanto, where he was the head of their global microbials discovery site. And Nick Macris is our head of business development. He was at Marrone in their early days through to their IPO. He's the one who's working with us to secure all of the distribution deals that we're after. I'm not going to belabor the advisors we have, but I do want to say a couple words very quickly. Adam Arkin is the inventor of our core IP. He's a brilliant scientist at the Berkeley National Lab and UC Berkeley. Erkki Aaltonen joined our board of directors from Yara. Anna Rath and Matt Ashby have great industry experience that they share with us freely, and they provide a lot of value to the company. And so with that, I'd like to thank you all, and I look forward to some discussion and some questions. Oh, and I'll also add, you can see my email address there. Please do feel free to reach out.
That's perfect. Thanks, Jamie. We do have a couple of questions from the audience, and we'll continue to field some more here. But the first one, the first two, I'll start with the first one. Do you see Join Bio, the JV of Ginkgo and Bayer, as a potential competitor, and where do you see Boost's key points of differentiation?
Yeah, absolutely. I would actually see Join as a great potential collaborator. Now, what I understand of Join, of course, I have no inside knowledge. What I understand of Join is that they're leveraging Ginkgo's platform to make to use genetic engineering and synthetic biology to make all kinds of wonderful agricultural products. When they announced themselves, they were particularly focused on nitrogen fixation. I would actually see Boost's technology as a wonderful complement to what they're doing. And I'm gonna zoom that out for just a second. If you think of synthetic biology, whether it's Joyn or any of the, or Ginkgo or any of the number of other wonderful companies in this space, if you think of synthetic biology generally, their unit of operation is the gene. They'll take some organism, they call it their chassis, and then they'll swap in whatever genes they think are appropriate. They'll remove genes, they'll change promoters, et cetera, but they're always usually operating within the one chassis. Boost's underlying technology has to do with understanding how microbes actually work together. So rather than synthetic biology, you can think of us as synthetic ecology, where the unit operation is the cell, not the gene. So this is where I'm going with saying that we could be a wonderful collaborator for companies like Joyn and many other companies in the space, where they might have a highly engineered chassis organism, but Boost technology would provide a wonderful supporting ecosystem for that engineered microorganism. So those two technologies would go very well together, and they'd play very well in developing new products. So thanks. It's a very interesting question.
Excellent. Thanks, Jamie. The second question is, could your technology approach work against, I might butcher the name of this bacteria, Aspergillus flavus and Aspergillus parasiticus mold infections?
We think absolutely. Now, I'm going to distinguish something. On the one hand, does our lead product work against Aspergillus? The answer is we don't know. It certainly could. It's certainly worth testing. But then sort of the separate part of the question is, if our lead product doesn't, could we develop a new product that does work against Aspergillus? I think there's no reason why I wouldn't think that. Absolutely, I think that would be a great target. We're certainly thinking about the applications to prevent aflatoxin, which I suspect is where you're going with that question. Maybe not, but that's my suspicion.
Mm-hmm. And in terms of on the regulatory side, what type of product registration is Boost Biomes planning on seeking?
Yeah, so we're seeking biopesticide registration through the EPA. If you think about the chemicals registration and the biologicals registration, these are very, very different animals. Chemicals, as many people know, that's a multi-year, probably hundreds of millions of dollars proposition. With biologicals, it's 18 months once we submit our data dossier to the EPA. It takes about 6 to 8 months to develop that dossier of data to submit. And so it's a much more straightforward process. Biologicals are typically thought of as being fairly safe. The EPA is looking for environmental safety, as you would expect. We've already done the first study on rats and showed that it was completely safe. There was no signs of toxicity in rats at the highest single dose. There are certainly a lot of additional studies to be done. And I see also that there was a question like this typed into the chat box, not the Q&A box. And so, since these are tied together, I'll answer that as well, which is, the effects of this in the human body, first of all, we think is a very, very low likelihood of it being any health issue, because first of all, they're not chemicals, they're biologicals, but some of these strains that we work with are already known in the food system to some extent. They're already used in some cases. In some industrial processes. So we think that it's a very low likelihood that they're going to have any kind of consequential effects on humans. Of course, we're going through all of the regulatory requirements, and obviously we're going to only bring these products to market as being safe.
Got it. Jamie, are there any like unique manufacturing challenges to your product? So when you think about producing biologics, there's always questions about scale up. There's always questions about contamination. Is it pretty straightforward for you from a standpoint of producing a microbial product, or is there anything unique here?
I don't think there's anything unique. I would say again, this comes from working with such great partners that we have in manufacturing. They've grown our product many times at sort of lab scale fermentation, 14 liters. They tell us that once they've got a repeatable process at 14 liters, they can scale that effectively infinitely. But actually, one thing that's really interesting about our product, David, I'm really glad you asked this question. One of the things that's really interesting, generally speaking, when you have a multi-microbial product, most people would think to ferment those products separately and then mix them into the final formulation. Our partner has actually found that the fermentations themselves are more effective if you ferment together, meaning that we get more product out of the fermentation when they're together versus if you ferment them separately.
And is that something to do with the symbiotic relationship between the two organisms, or is it something different? Do you expect to see that more going forward with other products?
Yeah, I can only speculate. I speculate that the answer is yes, but we do not have hard data on that. Yeah. And so I will not give a firm answer, but that is the sort of the obvious hypothesis.
Okay, got it. One further question we have from the audience here. Could the platform technology work outside ag tech? So i.e. in the skin microbiome, in hair, underarm, sort of other human health oriented microbiome applications? Absolutely.
We fully expect that that's the case. I mentioned Adam Arkin, who's the inventor on the core technology. He first published, the first was looking at wastewater. Ultimately might be used for wastewater remediation, but it was really just to understand the microbes in wastewater. So we absolutely expect that the platform is very broadly useful. Yes.
Got it. Okay. Grow, looks like we got another one coming in here. So in terms of the product application, when is the application made? Is it made shortly after harvesting? How is the product applied to a crop? Is it difficult to make a thorough application? Can over-application be an issue?
The last of those questions, over-application, we don't know. So I'm gonna leave that as not answerable. But interest. And so if it's
Jamie,
sort of a
Jamie, we lost you there for a couple seconds.
So have you got me back?
Yep.
Okay. All right. And I see that Kent was asking the question and added before harvesting. Yeah. Application is made on sort of a normal schedule for crop protection. So if you're, grapes, 8 applications over the course of a season, it would be 8 applications, or one of the treatments used over the course of those 8 applications. Let me see. I'm just parsing out that question. Yeah. So I think it's, as of now, we would recommend it as sort of a regular treatment schedule. But again, one of the suspicions we have about the usefulness of our platform is if our microbes truly are working together and they're really benefiting from each other, we suspect that we should be able to do less frequent treatments than what farmers normally do today. Again, I'm gonna be transparent. That's a speculation today, but we think that there's good reasons to think so. Thanks for the question.
Great. Well, I'm going to pause here and see if there's any further questions from the audience. So if you do have anything, feel free to fire away. Another question, do your proprietary organisms remain resident in the soil, and do future crops benefit from past applications of those microorganisms?
Yeah, it's a great question. It's on our plate to do that work. We haven't done that. It's a really interesting question.
Yeah, it's certainly interesting. Well, Jamie, one thing we always like to ask our guests at sort of the end of the Q&A section, make sure there's nothing else coming on here, is what can the audience do to help you out here? Where does Boost Biomes need the most help?
Sure. And there's like any startup, I think there's lots of ways we can use help. First, in the sense of co-developing new products, there were some questions about that. Questions about applications in different areas or applications in agriculture that we haven't tackled yet. Certainly, we're happy to have those discussions. I mentioned earlier that we are very actively pursuing distribution for the product in other crops within the US or in other geographies. Folks that have expertise, either that have a business interest in, around that level, expertise in regulatory requirements outside of US. So that as we're starting to think about that, we'd love to have those discussions. Really, there's a lot of breadth and a lot of different ways. Interesting. I'd encourage you to reach out. We're always happy to have at least a discussion.
Perfect. Well, Jamie, thank you for joining us today, and congratulations on all the progress to date. I'd also like to thank the audience for their active participation today. A lot of really great questions from everybody, really thoughtful, meaningful questions. To let you all know, for anybody who's new to the webinar today or listening to this on a recording, we host these calls every week at 3 p.m. Central. You can register for every week on Thursdays at 3 p.m. Central. You can register for the Agrifood Conversations webinar series by going to agrifoodconversations.com. If you know anyone that would like to watch this webinar, feel free to share it with them. A replay of the webinar will be emailed to you in the next 24 hours, and they can view it on agrifoodconversations.com as well. But otherwise, Jamie, thank you so much. Congratulations on all the progress. Thanks to our audience for joining today, and we look forward to seeing you next week.
And thank you to the iSelect team. Appreciate it.
Thanks, Jamie. See you. Bye, everybody.